There are many issues with urbanization, one of them being fragmentation of ecosystems. This can be seen in the picture on the right, where the ecosystem is on the left, whereas urbanization, or the concrete of the outflow stream, on the right. This can also lead to passage challenges for the organisms, and both of these situations can cause a change in organism abundance. Passage challenges are things such as there are too many grasses within the stream, and this can lead to limited resources such as less habitats and less organisms, or sunlight, depending on how much vegetation there is. However, based on this urbanization, it can also serve as a model system for social and biophysical patterns and processes. This could be an example of, on a small scale, a stream, which would look like with urbanization, but on a large scale could be something much larger. However, urban areas are becoming increasingly hand in hand with wildlands, and this is leading us to know the understanding of aquatic systems, both all physically, biologically, and chemically throughout this study that we have performed.
The purpose of this lab was to better understand aquatic ecosystems by running several tests related to stream health and by collecting benthic organisms living in these aquatic systems. These data would be compared with previous year's data to see how these systems are changing in response to time.
We hypothesized that we would find differences regarding the chemical composition of our stream section and the benthic organisms cataloged there when comparing our data with data collected in previous years. According to A.U. Urvinia in her journal article, Nitrogen Sources, Processes, and Associated Impacts of Climate and Land Use Changes in a Coastal China Watershed, increased urbanization and emissions cause nitrate levels to increase as there is a positive relationship between amount of fossil fuel combustion and nitrate levels found in urban streams. Urvinia also argues that there is a strong positive correlation between nitrate levels and algal bloom. With an increase in algae, there's typically an increase in organisms to decompose dead algae, as referenced in the journal article, An Innovative Index for Evaluating Water Quality in Streams by Ahmed said. This article went on to state that these organisms will deplete dissolved oxygen in the water. With decreased oxygen levels, the temperature of a stream can increase, which can have a negative effect on organisms living there. Therefore, it was hypothesized that when comparing our data to years prior, we would see an increase in nitrate levels, a decrease in dissolved oxygen, and an increase in temperature, which would therefore impact the different taxa that were observed in our stream section.
To accurately collect and record other measurements in the lab, the stream area was first defined by performing a series of measurements. First, a 5-meter section of the outflow was marked with flags. Then, at 5 equal intervals along the stream cross-section, the depth and distance from shore was recorded using a tape measure and meter stick. The distance was measured from the same point on the shore for each section.
To measure the velocity of the stream, a small plastic cap was turned upside down and placed in the stream at one end of the 5-meter section. Once it was released, a timer began timing until the cap reached the end of the 5-meter section, and this was performed 3 times. In order to accurately measure the discharge of the stream, a cross-section of the stream was chosen. Perpendicular to the flow of the stream, the depth was recorded at 3 different points across the chosen line, and this data was later used in the calculation of discharge.
From the water collected at the outflow, chemical tests were taken back at the lab. The picture on the right shows what this would look like. To find the phosphate and nitrate levels, an indicator molecules were added to the water that had either phosphate or nitrate, shaken to cause the reaction, in which turned the water pink, whether to show the phosphate or nitrate levels. To find the concentration value, the spectrometer was used to show the milligrams per liters after the waiting period of 15 minutes to find the reaction between the water and the indicator molecules.
A vernier probe and software was used to take measurements regarding temperature, pH, and dissolved oxygen. First, the stream was divided into 3 subsections, upstream, midstream, and downstream. To measure the pH, a pH probe was connected to the vernier lab quest, and the tip of the pH probe was rinsed with DI water before being placed in the stream in the 3 different subsections. The pH probe was placed in the water for approximately 45 seconds before the pH value was recorded. This was to give the probe enough time to stabilize and ensure that the reading was accurate. The pH results for the 3 subsections were recorded, and from the results an average was calculated. This procedure was repeated with both the temperature and dissolved oxygen probes, where once again the data was recorded for each subsection and an average was calculated.
Another portion of this lab was dedicated to collecting benthic organisms and categorizing them by taxonomy. To collect benthic organisms, we took a net and aggressively agitated the water in our stream section, and then we scooped up all that mud and all that water with the benthic organisms in it and transferred that to a collection jar. We then took that collection jar back to the lab and categorized them by taxonomy.
The average depth of the cross-section was taken 5 different times, and that was averaged out to be 0.1432 meters. This was then multiplied by the width of the cross-section, which was measured out to be 2 meters wide. To find the area of the cross-section, we multiplied those two numbers together and got a total of 0.2864 meters squared.
3 values of flow velocity were averaged to yield an average velocity in the outflow stream section of 0.0683 meters per second. The discharge was calculated to be 0.00978 meters cubed per second. Using the Vernier software and probe, the temperature, pH level, and dissolved oxygen values were recorded. After taking 3 measurements of each, 1 downstream, 1 upstream, and 1 in the middle, the average was calculated for the 5-meter long section of the outflow. The raw data used in these calculations is located in Table 4 in the appendix. The average pH was calculated to be 7.513. The average surface temperature was calculated to be 15.3 degrees Celsius. The average temperature 1 centimeter from the bottom of the stream was calculated to be 15.1 degrees Celsius, and the average dissolved oxygen was calculated to be 6.90 milligrams per liter.
After testing the water chemistry using the Hatch water test kits with handheld spectrometers, the phosphate level was found to be 0.23 milligrams per liter, while the nitrate level was found to be 1.11 milligrams per liter. In 2019, the phosphate level was 0.27 milligrams per liter. In 2018, the phosphate level was 0.3 milligrams per liter. And in 2017, the phosphate level was 0.34 milligrams per liter. In 2019, the nitrate level was 0.26 milligrams per liter. In 2018, it was 0.25 milligrams per liter. And in 2017, the nitrate level was 0.29 milligrams per liter.
Abiotic material within an ecosystem is material that is non-living, while biotic material is living or once-living material. We use data collected from biotic material at this site to determine the Shannon-Wiener Diversity Index of 1.41. The Shannon-Wiener Diversity Index is a commonly applied measure of species diversity that uses both richness and evenness to determine a value.
The following table presents the identified order of organisms collected and the estimated number of each order in the sample. The orders here are the ecologically functional groups. More discussion will come on this later.
The graph on this slide shows the estimated number of organisms in the collected sample separated by the order of the organisms. It is showing data from both 2019 and 2020 in order to compare these 2 years. More will be discussed on this later.
From the phosphate level results, there is a 1.17 decrease from last year and a 1.5 decrease from 2017. Overall, phosphate levels survive at a temperature degree of 25 degrees Celsius or lower. This could be the reason for the phosphate to be in the river to begin with, as seen from the table that showed that the water temperature was 15 degrees Celsius. In terms of why there was a decrease, it could have been from the lack of runoff from highly developed urban areas from this park, such as less walking around and less things being contaminated into the river. Also, this could be from different types of and coverages of vegetation, such as the types of vegetation within the stream caused phosphate levels to be decreased, as well as organisms that eat the certain bacteria that contain phosphate.
The nitrate level collected in our segment of the stream was found to be 1.11 milligrams per liter. This was a dramatic increase from the years prior, as between 2017 and 2019, the nitrate levels only varied from 0.25 and 0.29 milligrams per liter. According to A.U. Urvenia in her paper, Nitrogen Sources, Processes, and Associated Impacts of Climate and Land Use Changes in a Coastal China Watershed, these increases in nitrogen can be caused by increased fertilizer use, runoff from nearby residential areas, and fossil fuel combustion. It's possible that these sources are causing this dramatic increase in our stream nitrate levels, as these would be expected with climate change and the fossil fuel combustion, and as Denver is urbanizing, more fertilizers would be typically used. However, this is a pretty dramatic increase. We also noted that we had disturbed the stream prior to water collection by stepping into the stream, so it's possible that we collected a bad water sample, and this is why we're seeing this dramatic increase. If we were to perform this experiment again, we would definitely want to rerun the nitrate levels.
Now, to continue on the discussion aspect, we're going to talk about the abundance curves that Olivia showed earlier. We determined that it would be useful to perform a statistical test on the very minor difference between the amphiphota populations between 2019 and 2020. Now, when observing the large difference in Diptera species, we can assume that it's reasonably significant and possibly explained by an increase in nitrates. Of course, we would have to also perform a statistical test, but we can observe that it's most likely significant given the massive difference. We can also note an emergence of Platyhelminthes and Hemiptera species, given that there were none counted in 2019. And we believe that the possible uptake in the species for Diptera, Platyhelminthes, and Hemiptera has to do with an increase in nitrate composition in the water, which could be products of urban runoff or agricultural runoff, possibly. Okay, so the final section of discussion will be what we could possibly change about our experiment, if anything. It's possible that we could redo some of the later nitrate samples that we took because there was a bit of sediment disturbance that could have affected the measurements of nitrates. The stream section we had to perform the width and flow tests had a lot of obstacles, and it's possible that we would have a more accurate measurement if it was unobstructed and the water could flow more freely with the cap on top. So the real takeaways that we got from this experiment is that there's a heavy effect on biodiversity in urban aquatic ecosystems that occur from urban runoff and various human activities that can affect the nitrates and other biotic and abiotic factors. We also discovered with that large increase in Diptera species that Dipteras could be more adapted to high nitrate environments. So a possible future experiment that we could do would be testing two separate locations, one high in nitrates, one low in nitrates, with a lot of other controls in order to determine what the significance in the difference between Diptera species actually are.